Thank you so much, Meg, and good afternoon, everyone. Thank you so much as well for your patience to stay until this last talk before we going for dinner tonight. I'm going to talk about the project we have been doing in the past couple of years, including two projects. One is try to develop these high moisture retention materials. The other one is to develop this alternative microbial carrier material, particularly for rhizobium inoculants. So I'd like to thank, before I forget, I'd like to thank our research partners, industry partners, our team members. This is basically is a very big team. This is a very big teamwork, okay? Thank you so much. So I like to talk about this a little bit background information. We have, in Australia, we produce probably 10 million this waste, we call pre-farm gate waste each year. At the same time, nationally, we produce 14.6 million tons of organic waste. About 47 currently being recycled to produce many compost or organic amendment, and 9% for energy generation. About 44% of this organic waste currently go to landfill. If we have this alternative to divert this 44% organic waste from landfill, that will create about $1 billion industry. Okay, this basically product would be compost only. Another issue with waste industry, the majority of this product we produce are compost, probably 95%, and the value is very low, about from $80 to $250 per ton. On the other hand, we in Australia we have historical problem is a drought, periodical drought. It is big issues that constrain the big constraint in the farming system. They cause this low lower germination rate, seedling survival rate is low as well, and at the same time, this rhizobium survival rate is very low. It is critical for the first months to make sure this rhizobium can survive, can form the symbiosis with the host plant, and to form the nodule, otherwise they will die. So this is background information. So what I talk with basically is going to summarize this, the two projects. So what we did in the past couple of years, we collected about over 100 different material, and including organic and inorganic materials, clay materials, and we characterize these materials. Okay, then we do this functionality test, and we optimize this formulation for as a rhizobium carrier. Then we did a pod trial, and the control environment trial, and a field trial. On the other hand, we in terms of high moisture retention materials, so we use this organic waste. In this case, we try the tree bark. There are some crop residues. Use this cross-linking methodology to link organic functional group with the polyacrylate, and to increase this high moisture retention this capacity. These are some results. So overall we find there is a few materials there: meal mud, peanut shell, diatomite, and sugarcane bagasse, and mulch, and they have quite high the structures material and large surface area, and the other thing, the large amount of functional group on their surface, basically they contribute to the high moisture retention capacities, and this rhizobium carrier, is a very good habitat for rhizobium bacteria. In terms of formulation, meal mud, diatomite, and cow manure and compost, and then low temperature biochar. The inoculants formulation has great viable cell, microbial cells, and survival rate, and the long life, share life. So we also did this there's long share life, 1 year studies. The Mulma-based formulation of greatest share life, okay? Biochar-based formulation and the diatomite-based formulation, also they show quite good viable microbial cell counts as well. So in terms of potential novel organic-based high moisture retention materials. So what we did, you can see from here, we use tree bark and we grunt. Then we use cross-linking methods. Then bring this organic functional group and this polyacrylamide together. In case of tree bark, palm tree bark, the moisture retention capacity increased by 30 times, okay? So this material was being tested in the glasshouse trial and pot trial, show that its moisture retention capacity increase, and also the increased seed germination rate, and also is eco-friendly, improve soil health because there's organic, about 35% organic materials there. But this material need a further, this large-scale glasshouse trial and a field trial. Next, I'm going to talk about this glasshouse trial and field assessment of alternative rhizobial carrier products here. So this is closed chamber experiment. Basically, is this control environment, the temperature, moisture, and it's all controlled. We use vermiculite as growth medias. So you can see quite clear here, we use a different control, uninoculate control, peak control, then also we compare the 5 formulation. 4 of them are the seed coating material, the other one is granular material. So I showed some of the result here. You can see in this biomass. Driver, you can see very clearly, Mulma formulation and the biochar, low temperature biochar formulation have higher biomass, okay, compared with other controls. So in terms of inoculation and the nitrogen fixation, you can see very clearly here, the same trend, there's diatomite formulation and biochar formulation have greater nodule numbers and the nitrogen fixation rate. To summarize, results from the growth chamber experiment, so the soybean and chickpea plant growth, the Mulma is better than biochar and the diatomite-based formulation. For lentil and field peas plants, biochar-based formulation is better than other treatment. So overall, Mulma, the formulation, Mulma, the biochar formulation, and the diatomite formulation have the potential to be used as alternative rhizobial carrier materials. In terms of greenhouse trial, we tried the 4 seed coating products, okay, and the 1 granular product. We also have control without inoculants, and peat inoculants, and also 1 commercial products. Control the moisture at the 2 levels. 1 is drought, stress. The other one is optimum water holding capacity. We carried out in the 2 different soils. So you can see it's quite clear here. Mulma formulation and low temperature biochar also have this higher biomass. The similar result for nitrogen fixation, ethylene production, it is indicator of nitrogen release activity of nodules. Okay, so you can see quite clearly this Mulma formulation and the biochar formulation have higher the nitrogen fixation. to summarize from the greenhouse trial, Milma-based formulation outperformed the peat and other carrier formulations, show quite promising result. Biochar diatomite-based formulation could be also used as potential rhizobium carrier formulation. So cow manure, seed coating, and granular is comparable to other to peats and commercial product. So finally, I'm going to talk about the field trial. We carry out the 4 field trial. The 3 of them already finished. So the field peat trial was in this WA, carried out by David Minky's group. Chickpea was in this central New South Wales, the central farming system, New South Wales. Lentils was carried out in South Australia. So in terms of the result for field pea and the field trial and the chickpea field trial, Milmark and biochar and the diatomite formulation showed a better rejuvenation and harvest index in years, but the difference is not statistically significant. So this is just indicating the spatial variation, in the field, and it's not easy to get a consistent result. We need to have more field trial. But the trend we can see quite clearly is Milmark, low temperature biochar, and diatomite, the material that got a potential to be used as these alternative carrier materials. To conclude, so from the past couple of years experiment, our research showed that great potential to be able to use this recycled organic to create a value-added product for circular farming systems, particularly in our Australian environment. Milmark, low temperature biochar, and diatomite are excellent materials as alternative microbial carrier materials for effective inoculation formulations. Recycled organic could also be used to create eco-friendly, high moisture potential retention materials for building drought resilience farming system. So in terms of high moisture retention material, they think further work needs to be done. So next step, what we do, we're thinking about 2 things we can do. One thing, we actively engage with potential commercial partners to see if anybody interested in that. We already talked to a few partners. We're still engaging. The other one, I think because we test over 100 different materials, this material, most of them are locally available and also affordable. The farmer can use this material for their, the routine use, for example, as rhizobium carrier materials. Okay, for example, they can use Milmark, can use biochar, as the carrier material. They can mix with this freeze-dry, these inoculants for their use. I think that's all from me. Okay, any question? I'm happy to answer. Thank you so much.
Thank you, Chen. This is one of the great examples in Program 3, developing materials in the laboratory, taking to the field implementation at multi-locations in different states. So I think there's a time only, quick, two questions. Yeah.
Absolutely great work from lab glass house to field study. I remember working with Milmut many years ago, and I saw a huge difference between Milmut and other amendments, and that difference was directly linked to very high labile nutrient content of Milmut. So in your study, when you look at all the other amendments, for instance, per unit gram of Milmut, I believe nutrient content will be fairly high. So in your study, to compare the different amendments, well, I haven't seen, you said, mentioned this, without looking for consistency in nutrient content, it will be difficult for me to judge the differences in yield. Because if I was to do that, I would have looked for nutrient content per gram, and then I would have asked this question, what difference will it make if I was to provide the same nutrient content to all the materials and then see the difference? You might not agree with me, but just I'm putting this to you.
Actually, it is a very good point because the reason actually for the meal inoculants, I thought it's not a nutrient, it's active carbon, biological carbon for this rhizobium. But you talk about this meal mud grows better. But in this case, the quantity of meal mud, all this material is very small. For seed coating, it's nothing at all. I don't think they provide a substantial difference in terms of nutrient inputs. It's carrier material, it's not organic amendment. So it's a different story. Okay, thank you.
Regarding the biochar, did you just try the pine bark or did you try other biochars?
So the biochar, in this case, we use a low temperature pine wood biochar, about 450 degrees. Actually, in that biochar, pine wood biochar, it was produced in WA here. Okay, actually we shipped back to Brisbane a couple of years ago. For the pine bark, we used to create a high moisture retention material. Okay, for the biochar, we use pine wood.
Okay, but from pine, not from other sources?
Because of biochar, we collected over 120 different type of biochar in our lab. We tested, you know. Just biochar, we also created biochar under different temperature, use different feedstock. They are all different, okay? They have different properties. So the problem, you know, when I talk about biochar, you know, I always remind myself, you know, biochar, this biochar is different from other biochar. Although they all call biochar. For example, we use a cup. You can make up out of glass, can make up different material, plastic, you know. It's not the same thing. So in this case, we try different material. Low 450 degree, create pyrolysis temperature, create a biochar from pine wood. They are the best one. They have a large surface area. They have this polar structure, fiber structure.
Yeah, thank you, Chen. Chen is